On this show, we spend a lot of time talking about systems. We talk about the nervous system, how it's like this incredible computer network, or the digestive system, which is kind of like a factory.
Right, these big, complex machines.
But there is one system that connects, well, it connects literally everything else. It's the river of life.
Mm.
And I don't mean that in some, poetic way.
No, not at all. It's quite literal. If you stop the flow of that river, the game is over in, a few minutes.
It's the logistics network, the sewer system, the heating grid, and the standing army, all rolled into one red liquid. We were talking, of course, about blood.
But not just blood in the way we usually think about it. I think when most people picture blood, they picture the traffic. They're thinking about the red blood cells carrying oxygen, or maybe the white blood cells, chasing down a bacterium.
Right. Yeah, we picture the cars on the highway. That's
Yeah.
a perfect analogy.
Exactly. But today, we are going to completely ignore the traffic. We're looking at the road itself. We're doing a deep dive into the material from Chapter 52, specifically focusing on plasma proteins and immunoglobulins.
We're zooming in on the invisible stuff.
The invisible, dissolved infrastructure that allows all that traffic to move in the first place.
Okay, so sticking with that highway analogy then, if the cells are the cars, the things we're discussing today are, the tow trucks, the police officers, the patch crews fixing the potholes.
And the hidden cargo, the stuff strapped to the roof of every single vehicle.
I like that.
That is a perfect way to frame it, because without these plasma proteins, without this liquid infrastructure, the cells would just be stuck. They couldn't flow. They wouldn't have the right pressure. They just wouldn't survive.
We have a massive stack of notes here. We're going to cover everything from, and I'm quoting here, the sponge that keeps your blood from just leaking out of your veins.
Which is an amazing concept.
All the way to the heist movie, that is your body's iron recycling program.
That's my personal favorite section. I mean, the lengths the body goes to not lose a single atom of iron is just staggering. It's a true economy of scarcity.
Now we're going to get to the heavy artillery later on, the antibodies. But I want to start with a provoke. The notes mention a specific connection between smoking and lung damage. But it's not what I thought it would be. It's not just smoke irritation.
No. It's so much more precise than that. It is a molecular assassination. We are going to learn exactly how cigarette smoke chemically targets a single amino acid on one specific protein, and how that one tiny chemical change just completely destroys your lungs' protection.
Wow.
It is one of the clearest examples of biochemistry explaining a disease that I have ever seen.
And that's really the why it matters, isn't it? Okay, so let's start at the very beginning. The liquid itself, the foundation.
The plasma.
Right. So, to get our bearings, if I take a test tube of blood and I spin it down in a centrifuge, what am I actually looking at? What are the layers?
So gravity, or in this case, the extreme G-force of the centrifuge, it's going to pull all the heavy stuff down to the bottom.
Okay.
That's your red blood cells. That dense red plug at the bottom of the tube is called the hematocrit. In a healthy person, that's about, say, 45% of the total volume.
And sitting right on top of that red plug.
There's a very thin, kind of whitish layer. It's called the buffy coat.
The buffy coat, which always sounds to me like a vampire hunter's jacket.
Well, you're not totally wrong, I guess. It's the white blood cells and the platelets, the body's defenders. But the top half of the tube, roughly 55% of the volume, is this clear yellowish liquid. That is the plasma.
Okay, and this is why I always get tripped up on the terminology. Plasma versus serum. I hear doctors use them almost interchangeably sometimes, but the text makes a really hard distinction.
And it is a critical distinction, especially for the lab. It changes what you can actually test for. So think of it this way. Plasma is the blood as it flows in your body. It is liquid blood, right? Which means it still contains all the proteins needed for clotting, specifically a big one called fibrinogen.
Okay, so plasma is the liquid component plus all the clotting factors.
Correct. So to get plasma in a test tube, you have to add an anticoagulant, a chemical that stops clotting, before you spin it down. Otherwise, nature just takes over.
And nature's first instinct is always to clot.
Exactly. If you cut your finger, or if you just put blood in a plain glass tube with no chemicals, that fibrinogen activates, it turns into fibrin, it creates this solid mesh that traps all the cells, and that's a clot.
So if I just let the tube sit on the counter and clot?
The liquid that's left over, the liquid that literally squeezes out from that clot, that is serum. So mathematically, it's pretty simple. Serum equals plasma minus the clotting factors.
Got it. Okay, that's clear. Serum is what's left after the clotting party is over.
Exactly. And serum is cleaner in a way for certain tests because all that fibrinogen is gone. But for our purposes today, we're interested in the whole universe of proteins floating in that liquid, whether it's plasma or serum. And it is a very, very crowded soup.
How crowded are we talking?
I mean, extremely. There are thousands of different proteins. And to sort them all out, scientists use a technique that sounds complicated but is actually quite elegant. It's called electrophoresis.
Which sounds like a sci-fi interrogation technique, but I know it's a standard lab test.
It's basically a foot race for proteins. Imagine a running track made of cellulose acetate. It's just a porous strip of material. You put a single drop of plasma on the starting line.
Yeah?
Then you hook up a battery. You put a positive charge, an anode, at the finish line, and a negative charge, a cathode, back at the start.
And because proteins all have their own electrical charges, they start to move.
Right. Most of the plasma proteins are negatively charged at the body's pH. So naturally, they want to run toward that positive electrode at the finish line. But, and this is the key, they don't all run at the same speed.
It depends on their size and charge.
Exactly. Their charge density and their size. So after a few minutes, they separate out into these distinct bands. It looks like a leaderboard from a race.
And who is the Usain Bolt of this protein race? Who's always the fastest?
Albumin. It wins every single time. It's relatively small, and it has a very strong negative charge, so it just sprints toward that positive anode. It moves the furthest, forms the biggest, darkest band right at the front.
And then you have the rest of the pack just kind of trailing behind it.
Right. You get these groups, these fractions. Right behind albumin come the alpha-1 globulins, then the alpha-2 globulins, then the beta globulins. And finally, bringing up the rear, the slowest and heaviest group are the gamma globulins.
And spoiler alert, the gammas, those are the antibodies, right?
Correct. And there's a really important manufacturing distinction here that helps doctors diagnose things. The liver is the factory for almost everything else.
Okay.
Your liver pumps out the albumin, the alphas, the betas. It is churning out 70 to 80% of your total blood protein. It's a powerhouse.
But not the gammas.
No, the liver doesn't make antibodies. Those come from your immune system, specifically from plasma cells, which are mature B cells. And this is clinically huge.
How so?
Because you can look at that electrophoresis strip and instantly get a snapshot of the body's health. If someone has, say, liver failure, the factory is shutting down. So that albumin band, the fast one, is going to be tiny or faint.
The lead runner is falling behind.
Exactly.
Yeah.
But if someone has a massive infection or a cancer of the immune system, like multiple myeloma, the gamma band, the slow one at the back will be huge and dense.
So just by watching the race, you can tell which organ system is in trouble.
Precisely. It's an incredibly powerful diagnostic tool.
Okay, let's talk about that star product from the liver factory, albumin. The notes here call it the heavy lifter.
It is the main character of the bloodstream, no question. Albumin makes up about 60% of the total protein mass in your plasma. Your liver synthesizes about 12 grams of it every single day.
12 grams doesn't sound like a lot until you realize that's pure protein synthesis. That's like making a scoop of protein powder from scratch every day, just for this one molecule.
It's a massive metabolic expense. The text says about 25% of the liver's total protein synthesis capacity is dedicated just to making albumin.
What does it look like structurally?
It's shaped like an ellipsoid, so think of a rugby ball or an American football. It's a single long chain of 585 amino acids all folded up. And it's tough. It's really tough. The text highlights that it's stabilized by 17 disulfide bonds.
Disulfide bonds, those are the sulfur-to-sulfur bridges, right?
Yes. Think of them like internal cross braces or steel struts inside that rugby ball. They lock the shape in place. This makes albumin incredibly stable. It can survive big changes in pH and temperature. It's really built to last in the turbulent river of the blood.
So we have billions and billions of these tough little rugby balls floating around. What are they actually doing?
Two major jobs. The first one is all about physics. Albumin is the sponge.
The sponge. Okay, this brings us to a concept called osmotic pressure. Now, this is something I remember struggling with in school, so let's break it down really carefully. There's a tug of war happening in our blood vessels.
There is. This is all governed by something called the Starling forces. So imagine your capillaries, the tiniest blood vessels where the real business of feeding your cells happens. These vessels, they're leaky by design. They have to be. They have little holes or fenestrations to let nutrients out.
Okay, so the prey has holes in it.
Right. Now, your heart is a pump. It creates hydrostatic pressure. It is physically pushing blood against the walls of that pipe. On the arterial side of the capillary, the side coming from the heart, that pressure is high, and it pushes water, sugar, and salt out of those holes and into the surrounding tissues.
So water is basically spraying out of the hose.
Exactly, that's how you feed the cells. But here's the problem. If that were the only force at play, all the water would leave your blood. You'd pump yourself dry in minutes, and your tissues would just swell up like water balloons.
But we desperately need a way to get that water back in.
Enter albumin. Albumin is dissolved in the plasma, but it's a big protein. It's too big to fit through the little holes in the capillary. It gets stuck inside.
So the water leaves, but the albumin stays behind.
Exactly. And this creates a concentration gradient. Nature abhors an imbalance. Because there's now a high concentration of protein inside the vessel and a low concentration outside, it creates a pull. It's a chemical force that draws water towards the albumin. This is called colloid osmotic pressure, or oncotic pressure.
So the heart pushes out, and albumin sucks in.
It's a perfect cycle. It's so elegant. On the arterial side, the heart's push is stronger than albumin's pull, so fluid leaves. As the blood moves along the capillary, the pressure from the heart drops off. So on the venous side, albumin's pull wins. The fluid is sucked back into the vessel, carrying waste products with it.
That is elegant. But this also explains why severe malnutrition causes swelling, right? I remember seeing photos of children with kwashiorkor, with starvation, and they have these distended, swollen bellies.
That image, that is the tragic physical result of this physics equation breaking down. If a child is severely malnourished, they aren't getting enough protein in their diet. The liver runs out of the raw materials, the amino acids. It simply stops making albumin.
So the concentration of those rugby balls in the blood just plummets.
Which means the suction force turns off. The heart keeps pushing fluid out into the tissues, but there's no albumin to pull it back in. So the fluid stays out there. It accumulates in the abdomen. That's a condition called ascites. It pools in the legs, the face.
And so they look full, but they're actually starving.
Exactly. The belly isn't fat, and it's not food. It's just water. It's edema caused by the complete failure of the osmotic pump.
That is a stark, purely mechanical explanation for something so heartbreaking.
It really is. It shows you how biology is just chemistry and physics at play, with real-world consequences.
Okay, so job number 1 is the sponge. What is job number 2?
Job number 2 is the Uber driver. Albumin is a major transport protein. But unlike some proteins that are very specific, like a limousine for one specific VIP, albumin is more like an Uber pool. It picks up almost everyone.
Low specificity, but high capacity. That's the phrase from the text.
Exactly. It has these hydrophobic pockets, little greasy spots inside its structure where it can tuck away things that don't like being in water. It carries free fatty acids, which are your fuel. It carries calcium, steroid hormones, bilirubin, copper, tryptophan. The list goes on and on.
And drugs. That seems really important.
Oh, this is huge for pharmacology. Albumin loves to bind to drugs. Aspirin, penicillin, sulfonamides, warfarin, you name it. When you pop an aspirin, a significant chunk of it immediately gets strapped to an albumin molecule and carried around the body.
Does that deactivate the drug while it's bound?
Yes, for the most part. While it's stuck to the Uber, it can't enter a cell or bind to a receptor. So albumin acts as a reservoir. It releases the drug slowly over time, which can extend its half-life.
So wait, if I have low albumin, say I have liver disease, and I take the normal dose of a drug,
You could potentially overdose. Because there's less albumin to soak up the excess, the free active level of the drug in your blood spikes to a much higher level than expected. Doctors actually have to calculate this protein binding percentage when they're figuring out your dose.
it really is the buffer for everything, isn't it? Water, drugs, hormones.
It stabilizes the entire environment of the blood. It's the ultimate multitasker.
Before we leave the general proteins, we have to mention the alarm system. The text calls them the acute phase proteins.
Yes, the liver isn't just a factory running on a schedule, it's a responder. When you have inflammation anywhere in your body, an infection, a burn, a broken bone, your immune cells release signaling molecules called cytokines. The big ones are interleukin-1, IL-1, and interleukin-6, IL-6.
These are the chemical messengers.
They're the fire alarms. They travel to the liver and essentially scream, "We are under attack." The liver responds by completely changing its production line. It downregulates things like albumin, we don't need maintenance right now, and massively upregulates defense proteins.
And the most famous one of these is CRP, right? C-reactive protein.
C-reactive proteins, CRP. Its levels can jump 1,000-fold in a matter of hours. It's an incredible response. It binds to bacteria and fungi and helps the immune system target them for destruction. Doctors measure CRP all the time to answer a simple question. Is this patient inflamed?
It's the universal check engine light for the body.
That's a perfect way to put it.
All right, let's shift gears. We've established a liquid foundation. Now we want to look at the economy of the blood, specifically the iron economy.
Ah, yes. This is the best part, in my opinion.
I was reading the notes on this, and the word that kept coming to mind was stingy. The body is just incredibly, unbelievably cheap when it comes to iron. It hoards it like gold.
It has to. There's a fundamental paradox with iron. On one hand, you absolutely cannot live without it. It's the core of hemoglobin. It's what carries oxygen. It's involved in the electron transport chain in your mitochondria. No iron, you suffocate at a cellular level.
But on the other hand,
it is incredibly toxic. Free iron, if you just let an iron atom float around by itself in the cell, is a wrecking ball. It catalyzes a process called the Fenton reaction.
the Fenton reaction, that sounds ominous.
It is. It generates hydroxyl-free radicals. These are some of the most reactive molecules known to biology. They shred DNA, they tear apart cell membranes, they denature proteins. So the body has two absolute rules for Iron Club. Rule number 1, you must have iron. Rule number 2, iron must never be left alone.
It always has to have a chaperone, a protein bodyguard.
Always. And because it's so dangerous to handle, we don't like to get it from the outside world if we can help it. We prefer to just recycle what we already have over and over again.
Let's look at the numbers here. A typical 70-kilogram adult has, what, 3 or 4 grams of iron total?
About 3 to 4 grams. To give you a sense of scale, that's about the weight of a small male. That is all the iron in your entire body.
And out of that, how much do we actually lose per day?
About 1 milligram, just one.
That's nothing. It's a rounding error.
It's 0.03% per day. We lose a tiny bit from shedding skin cells or the cells lining the gut, but otherwise it is a completely closed loop. We absorb about 1 milligram from our food to replace the 1 milligram we lose. Everything else, all the massive amount of iron needed for making new blood, is recycled.
Walk us through that life cycle. Where is the iron coming from?
It starts with the red blood cell. These guys have a tough life. They get squeezed through tiny capillaries, battered by turbulence, and they have no nucleus, so they can't repair themselves. After about 120 days, they're senescent. They're old and worn out.
They're clunkers, ready for the scrapyard.
So the macrophages, these big eater cells, mostly in the spleen and liver, they recognize these old cells and they engulf them. They eat the car.
And then they strip it for parts.
Right. They break open the hemoglobin. There's an enzyme inside the macrophage called heme oxygenase. It pops open the heme ring. This releases 3 things: a tiny bit of carbon monoxide, biliverdin, which becomes the yellow pigment bilirubin, and the grand prize, the iron atom.
So now the macrophage has the iron. But remember rule number 2. You can't just toss it back into the blood.
No way. It has to be carefully loaded onto a dedicated transport truck. That truck is a protein called transferrin. But, and here's a chemical hurdle that the text emphasizes, the iron that comes out of the heme is in the wrong chemical state.
This is where the chemistry comes back to haunt us.
Iron has 2 main states in the body: ferrous iron, which is 5 tau plus 10, and ferric iron, which is 5(3) plus $. The iron released from heme is in the ferrous 5 plus tau state, but transferrin the truck, it only accepts ferric 5(3) plus $.
So we need to oxidize it. We need to steal an electron from it before it can be loaded.
Exactly. And this requires a specific enzyme to do the job. That enzyme is called ceruloplasmin.
And this is where the plot twist comes in. Ceruloplasmin isn't an iron protein. It's copper protein.
It's a beautiful blue, copper-dependent protein made in the liver. This is why biology is so wonderfully interconnected. If you are copper deficient, you cannot make functional ceruloplasmin.
And if you don't have ceruloplasmin, you can't turn the 5(2) plus into 5(3) plus LAR.
And if you can't do that, you can't load the transferrin truck. The iron gets stuck. It's trapped inside the macrophage.
This blows my mind. So you could technically be full of iron. Your macrophages are absolutely stuffed with it, but you are functionally anemic because the iron can't get out into the blood to be used.
Precisely. This is one of the mechanisms behind a genetic disorder called Wilson disease. In Wilson disease, the body can't handle copper properly. Copper builds up in the liver and the brain, causing horrible damage, but the plasma ceruloplasmin levels crash to near 0.
Wilson disease, that's one of the rings in the eyes, isn't it?
Kaiser-Fleischer rings. It's copper depositing in the cornea. If you look closely, you can see a golden brown ring around the iris. It's diagnostic. But functionally, these Patients have a completely chaotic iron metabolism because their copper handling is broken.
So you need copper to move iron.
All
Okay,
right.
let's assume we do have copper. The iron gets oxidized by ceruloplasmin, and it hops onto the transferrin truck.
Transferrin is the armored car. It's a beta globulin. It can grab 2 atoms of ferric iron, 2 Phe317 plus ions, and it clamps down on them incredibly tightly. It transports them safely through the plasma to wherever they're needed, which is usually the bone marrow, to make new red blood cells.
Now, I want to zoom in on the delivery process, the transferrin cycle, because it's not just a simple drop-off. The cell actually swallows the truck whole.
It's a very aggressive delivery system.
Yeah.
Let's say you are a young red blood cell precursor in the bone marrow. You are desperate for iron to build hemoglobin. You put a flag on your surface called the transferrin receptor 1, or TFR1.
It's a sign that says iron needed here.
Exactly. The transferrin truck, loaded with iron, sees that flag and docks with the receptor. The cell then performs receptor-mediated endocytosis. It literally wraps its membrane around the receptor and the truck and pulls them both inside into a little bubble called an endosome.
The truck is now in the garage.
Inside that garage, the cell turns on proton pumps. It starts pumping acid into the bubble, dropping the pH.
And the acid makes the iron let go of the truck.
That's it. The acidity changes the shape of the transferrin protein. It loosens its grip. The iron atoms fall off and are then pumped out into the cell's cytoplasm to be used. The empty truck, now called apotransferrin, remains stuck to the receptor inside the bubble.
Do they just digest the empty truck?
No, that would be incredibly wasteful. The cell simply moves the bubble back to the surface. Once it fuses with the outer membrane and hits the neutral pH of the blood again, the empty truck falls off the receptor and floats away to find more iron.
It's unbelievable. The truck is never destroyed. It's just unloaded and recycled over and over.
A single transferrin molecule can make that trip thousands of times in its life. It's a perfect system.
But what happens if we have too much iron? We can't just keep driving it around endlessly. We need a bank vault.
That bank vault is a protein called ferritin.
Ferritin. I see this on my blood test results all the time. It's a measure of iron stores.
It is. Ferritin is a protein masterpiece. It's a hollow sphere made of 24 separate protein subunits. It looks like a microscopic soccer ball. Inside that hollow core, it acts as a mineralization chamber. It takes that dangerous reactive iron and crystallizes it into a safe, solid mineral form.
How much can one of these soccer balls hold?
One single ferritin molecule can pack away up to 4,500 iron atoms.
4,500.
It's incredibly dense storage. And it's dynamic. If the body needs iron, it can dissolve that mineral core and release it. But here's the catch. If the storage gets too full, if you really overload the system, the ferritin itself starts to break down.
And this is where hemosiderin comes from.
Yes. Imagine a warehouse that is so full that the walls start to burst and the roof collapses. The ferritin shells degrade and clump together into these big, insoluble granules called hemosiderin. You can see them under a microscope.
And seeing that is a bad sign.
It's a definitive sign of iron overload. It damages the cells. If a pathologist looks at a liver biopsy and sees hemosiderin staining, they know that liver is drowning in iron.
Before we leave the recycling center, there's one more scavenger protein mentioned in the text, haptoglobin.
Ah, yes. Haptoglobin is the suicide squad.
Explain.
So usually old red blood cells are eaten whole in the spleen, but about 10% of them just break apart inside the blood vessels. This is called intravascular hemolysis. When they pop, they release all their hemoglobin directly into the plasma.
And that's a problem.
It's a big problem. Free hemoglobin is small enough to get filtered by the kidneys. It can clog up the delicate renal tubules and cause acute kidney failure.
So haptoglobin saves the day.
Haptoglobin binds to the free hemoglobin instantly. The complex they form is massive, way too big to pass through the kidney filter. It keeps the hemoglobin safely in the blood until a macrophage in the liver can come along and eat the whole complex. But in the process, the haptoglobin molecule is destroyed. It's a one-way mission.
That's why haptoglobin levels drop to 0 in cases of hemolytic anemia. They've all sacrificed themselves to protect the kidneys.
Exactly. If a doctor sees low or 0 haptoglobin, they know for sure that red blood cells are popping open somewhere in the bloodstream.
Which leads us perfectly to part 3, the boss, the master regulator. We've got the trucks, transferrin, we've got the bank, ferritin, we've got the recycling center, the macrophage. But who tells them all what to do? Who sets the speed for the whole economy?
A tiny but ruthless protein called hepcidin.
Hepcidin. I feel like this is a relatively new discovery in the grand scheme of medicine, right?
It is. We knew there had to be a master regulator for decades, but hepcidin itself was only identified fairly recently. It's a small peptide hormone that's made in the liver, and its mechanism is fascinatingly destructive.
How does it work?
It controls the exit doors. Okay, imagine the two main cells that have iron they need to release. The gut cells, or enterocytes, that absorbed it from food, and the macrophages that recycled it from old red blood cells. Both of them have a protein door called ferroportin. It is the only known way for iron to get out of a cell and into the blood.
So ferroportin is the gate.
Hepcidin binds directly to ferroportin and triggers the cell to internalize it and digest it in the lysosome.
So it doesn't just lock the door. It tells the cell to literally eat the door.
It destroys the door. So if hepcidin levels are high, there are no doors. Iron is trapped inside the gut cells and then gets lost when those cells are shed, or it's trapped inside the macrophages. The net result is that the level of iron in the blood drops.
So hepcidin is the system's brake pedal.
It is the brake, the master brake.
And why would the body want to hit the brake?
Two main reasons. First, if your iron stores are already high, the liver senses high iron and pumps out hepcidin to tell the gut, stop absorbing more, we're full.
Makes sense. What's the second reason?
Inflammation. And this is the provocative thought we'll come back to. Bacteria love iron. They need it to reproduce. When the body detects an infection, again, signaled by that same cytokine, IL-6, the liver, ramps up hepcidin production.
It's hiding the money from the thieves.
It's a scorched earth policy. It locks all the iron away in the vaults so the invading bacteria starve. This is what causes the anemia of inflammation, or anemia of chronic disease. Your iron levels in the blood are low, but you aren't deficient. You're just sequestering it.
That is genius.
Yeah.
But what happens if this regulator breaks? The notes talk about the HFE gene and the disease hemochromatosis.
This is a classic genetic disorder. The HFE protein acts as the sensor. It sits on liver cells and helps them determine how much iron is in the body. In hereditary hemochromatosis, the HFE gene is mutated and the protein is broken.
So the sensor's broken, the thermostat is busted.
Right. The liver is flying blind. It thinks the body is perpetually starving for iron, even when it's completely full. So what does it do with hepcidin?
It keeps it turned off.
Hepcidin levels stay incredibly, inappropriately low.
And low hepcidin means all the ferroportin doors are wide open.
Wide open, all the time. The gut absorbs the maximum amount of iron possible, 247, for decades. And since the body has no good way to get rid of excess iron, it just piles up. It deposits in the liver, causing cirrhosis. It deposits in the pancreas.
And you get bronze diabetes.
That's the old nickname, yep. The skin turns a bronze color due to pigment and iron deposits, and the pancreas fails, causing diabetes. It also destroys the heart and joints.
And the treatment is fascinatingly medieval, but it makes perfect sense.
Phlebotomy, it's just bloodletting. You literally bleed the patient on a regular schedule. By removing red blood cells, you force the bone marrow to pull all that stored iron out of the organs to make new ones. You drain the bank by forcing the body to spend the cash. It works perfectly.
Incredible. Okay, let's move to part 4. The police force. The antiproteinases.
This section is all about preventing friendly fire. Our body produces incredibly powerful enzymes called proteases. Think of them as molecular scissors. We use them to digest food or to have our immune cells chew through bacteria.
But scissors can cut anything if you're not careful.
Precisely. For example, our neutrophils, a type of white blood cell, release an enzyme called elastase to chew up invading bacteria. But elastase, as the name implies, can also chew up elastin, the critical, stretchy protein that makes your lungs elastic and able to recoil.
So we need a safety cap for those scissors.
That safety cap is a protein called alpha-dollar antiproteinase, which is also known as alpha-1 antitrypsin. It circulates in the blood and in the lungs, and its main job is to find any stray elastase and inhibit it. It finds the scissors and clamps them shut.
And if you don't have this protein,
you get unchecked destruction. The elastase goes rogue and just starts digesting the lung tissue day after day. This leads to severe early-onset emphysema, the destruction of the delicate air sacs in the lungs.
Now, here's the part that I promised in the intro. The molecular connection to smoking. We all know smoking causes emphysema, but the text explains the exact molecular reason, and it is so specific.
It is the literal smoking gun. It comes down to a single amino acid on that inhibitor protein. The alpha-1 antiproteinase has a specific methionine residue at position 358 in its sequence.
Meth358.
That methionine is the bait. It's the part of the protein that the allostase enzyme binds to. It's absolutely crucial for the inhibitor to work. Now, what is cigarette smoke full of?
Nicotine?
And chemical oxidants, billions of them. So when you inhale smoke, those oxidants travel down into your lungs, and they chemically attack that one specific methionine 358. They oxidize the sulfur atom on it, turning it into something called methionine sulfoxide.
And that one chemical change matters.
It changes everything. The methionine sulfoxide creates a bulky, misshapen group that completely ruins the shape of the bait. The inhibitor protein can no longer bind to elastase.
So the safety cap doesn't fit anymore.
It just bounces right off. So a smoker is essentially giving themselves a functional acquired deficiency of this protective protein. They are chemically destroying their own shield, even if their genes are perfectly fine. The elastase is then left free to digest their lungs for years.
That is terrifyingly specific. It's not just smoke is bad for you. It's smoke chemically deactivates this specific sulfur atom on this specific protein, which leads to this specific disease.
Science is in the details. And of course, for people who have a genetic deficiency to begin with, the so-called PIZA genotype, they get emphysema even without smoking, and if they do smoke, it's a catastrophe.
There's one more police officer mentioned here, alpha-2,2 macroglobulin. This one sounds different.
It's the Venus flytrap. This one is really cool. It's a huge protein, and it's a pan-proteinase inhibitor, meaning it can inhibit almost any kind of protease. It doesn't target just one.
So how does it work? How can it be so general?
It uses a physical trap. It has a bait region in the middle that's exposed. When any protease comes along and tries to cut that bait, SNAP,
The whole protein closes on it.
the whole molecule undergoes a massive conformational change. It collapses and physically wraps around the proteases, encasing it in a cage. It doesn't chemically kill the enzyme, but it physically prevents it from touching any other large proteins.
It handcuffs the criminals so they can't do any more damage.
Exactly. And then, once that trap is sprung, receptors on the liver and other cells recognize the new shape and clear the entire complex from the blood. Job done.
Brutal. I love it. Okay, part 5. Sometimes proteins don't fold right. We have to talk about amyloidosis.
This is a much darker topic. We've been talking about proteins doing their jobs perfectly, but sometimes proteins misfold. They adopt an abnormal shape called a beta-pleated sheet.
And these sheets are sticky, right?
They are very sticky and insoluble. They stack up on top of each other to form these rigid fibrils. Think of it like pouring concrete into a sponge. This stuff deposits between the cells in your organs, your heart, your kidneys, and it stiffens them and stops them from working. This collection of junk protein is called amyloid.
The notes mentioned a distinction between AL and AA amyloid.
Right. It depends on what protein is misfolding. AL amyloid comes from immunoglobulin light chains.
The light chains from antibodies.
Yes, the smaller pieces of antibodies. This usually happens if you have a cancer of the plasma cells, like multiple myeloma. The cancer cells are going crazy, pumping out huge amounts of antibody parts. They make too many light chains, they can't be cleared fast enough, and they start to misfold and stack up as amyloid.
And AA?
A amyloid comes from a protein called serum amyloid A, or SAA. This is one of those acute phase proteins we mentioned earlier. So if you have a state of chronic, unrelenting inflammation like severe rheumatoid arthritis that goes on for years, your liver keeps making SAA. Eventually, the concentration gets so high that it starts to precipitate out as amyloid, usually in the kidneys or liver.
So it's basically industrial waste piling up from a war that never ends.
That is a perfect and very sad analogy, yes.
All right, let's pivot to something more triumphant. Part 6. The heavy artillery.
The immunoglobulins.
The antibodies. Our old friends, the gamma globulins. Let's start with the basic structure, the classic Y shape. Everyone's seen it.
It's iconic. It's made of 4 protein chains. You have 2 identical heavy H chains, which make up the Y stem and the inner part of the arms. And then you have 2 identical light L chains, which are the outer part of the arms. They're all held together by those strong disulfide bonds we talked about with albumin.
And the Y has 2 very distinct functional ends, right? The tips of the arms and the stem.
Absolutely. The tips of the Y are the variable regions. This is the business end. This is where the antibody grabs onto the specific invader, the antigen. The shape of these tips is unique for every different antibody. It's the lock that is custom made to fit only one specific key.
And the stem of the Y.
The stem is the constant region. That part is the same for all antibodies of a certain class. It determines what happens after the invader is caught. Does it call over a macrophage to eat the complex? Does it activate the complement system? The stem decides the effector function.
Okay, here's the math problem that always stumps me. Our entire DNA has fewer than, 150 genes for immunoglobulins? But our bodies can make millions, maybe even a billion different antibodies. How do you get a billion unique outcomes from only 150 instructions?
It is the ultimate genetic slot machine. We don't have a separate gene for every single antibody. That would be impossible. Instead, the genes are chopped up into segments. Variable segments, V, diversity segments, D, and joining segments,
D.
and J. Think of them like 3 decks of cards. You have a deck of V cards, a deck of D cards, and a deck of J cards. During the development of a B cell, the DNA literally breaks and rearranges itself. It randomly picks 1 V card, 1 D card, and 1 J card and stitches them together to make a unique variable region.
That's combinatorial diversity.
Right, and if you just multiply the number of cards in each deck, you get thousands of possible combinations, but that's not enough to get to a billion. So the body adds another trick, junctional diversity.
The sloppy glue.
That's a great way to put it. When the enzymes stitch the V, D, and J segments together, they are deliberately messy. An enzyme called TDT comes in and adds or deletes random nucleotides right at the splice points.
So it's just adding random letters to the genetic code at the seams.
Yes. This completely changes the reading frame and creates amino acid sequences that have never existed before in the history of the universe. And this all happens before the B cell ever even sees an infection. It's all preemptive.
And then, even after the antibody is made and finds a match, it can fine-tune itself.
Yes. This is the last layer, somatic hypermutation. If a B cell finds an antigen that kind of fits its antibody, it starts dividing rapidly, and as it divides, it intentionally introduces mutations into its variable region genes at a rate that is 1,000,000 times faster than normal DNA mutation.
It's evolving in real time inside your lymph nodes.
It's a process called affinity maturation. It keeps selecting for the mutations that make the antibody bind tighter and tighter to the target. So by the end of an infection, your antibodies are significantly better and more specific than they were at the start.
That is just mind-blowing. We're genetically engineering ourselves constantly to fight off bugs. Okay, let's run through the 5 classes of antibodies. The acronym is GAME-DE.
IgG, IgA, IgM, IgD, and IgE.
Let's do a quick roll call. Who is IgG?
IgG is the professional soldier. It makes up 75% of the antibodies in your blood. It's a monomer, just one Y shape. This is your long-term protection. It stays in your blood for months or years after an infection. And crucially, it's the only class that can cross the placenta to protect a fetus.
Okay, IgA.
IgA is the bouncer. This is the secretory antibody. Its job is to guard the gates. It lives in your mucus, your tears, your saliva, and your gut. It's usually a dimer 2 Ys clipped together. Its main job is to stop bacteria from ever attaching to your mucosal walls in the first place.
Next up, IgM.
IgM is the brute, the first responder. It's the first antibody made in a new infection. It's a massive pentamer 5 Y shapes all joined together in a giant star. That gives it 10 binding sites. It's not very specific, but it is amazing at clumping bacteria together, a process called agglutination, and at activating the complement system, which we'll get to. It's clumsy, but very powerful.
IgD?
IgD is the introvert. It mostly just sits on the surface of B cells and acts as a receptor. It doesn't do much swimming around in the blood. Its job is to receive the initial signal and tell the B cell to wake up and get to work.
And finally, the famous one, IgE.
IgE is the specialist and often the troublemaker. IgE's original design was to fight parasites, like worms. It has a special tail that lets it bind to mast cells and basophils. When it sees its target antigen, it triggers that mast cell to degranulate, to explode with granules of histamine.
That's the boom of an allergic reaction.
That boom is meant to cause inflammation and mucus production to flush a worm out of your gut. But in the modern world, without worms, it usually just reacts to pollen or peanuts or cat dander. That's your allergy. That's anaphylaxis.
It's a powerful weapon system that's pointed at the wrong target.
Exactly.
Finally, that brings us to part 7. The kill switch. The complement system.
This is one of the oldest, most ancient parts of our immune system. Even primitive animals have versions of this. It's not a cell, it's a cascade of proteins.
It's like a row of dominoes.
Exactly. It's a series of about 30 inactive enzymes or zymogens floating in the blood, numbered roughly C1 through C9. When the first one gets activated, it cleaves and activates the next, and that one activates 10 of the next, and so on. It amplifies the signal massively.
And there are 3 different ways to push that first domino, right?
Yes. There's the classical pathway. This one starts when an antibody, usually IgM or IgG, binds to the surface of a bacteria. The first complement protein, C1, then comes along and binds to the stem of that antibody. It basically says, I see you've caught one. Let's start the attack.
Okay, what's the second way? The lectin pathway.
This one is more primitive. It doesn't need antibodies. It relies on a protein called mannose-binding lectin, or MBL. This protein is designed to bind to specific sugar patterns, like mannose, that are found on the surface of bacteria and fungi, but not on human cells. It just recognizes foreign sugar patterns.
And the third one, the alternative pathway.
This is the tick-over pathway. The C3 protein, which is central to the whole system, just spontaneously activates at a very low level all the time. If it happens to bump into a bacterial surface, it sticks and kicks off the whole cascade. If it bumps into one of our own cells, we have special regulatory proteins on our cell surfaces that immediately shut it down.
So all three roads lead to the activation of C3.
C3 is the nexus. It's the central point of convergence. The pathways all lead to an enzyme that cleaves C3 into two important pieces, C3a and C3b.
And what do those two pieces do?
C3b is an opsonin. It's a tag. It covalently binds to the surface of the bacteria, coating it. This is opsonization again. It's like putting flags all over the bacteria that scream to phagocytes, eat this now. It makes the bacteria delicious.
And the little piece, C3a.
C3a and a later piece called C5a are anaphylatoxins. They are chemical sirens. They float away from the battle and cause inflammation. They recruit more immune cells to the area, and they make the local blood vessels leaky so those cells can get out into the tissue.
But the cascade doesn't stop there. It goes all the way for the kill, the MAC.
The membrane attack complex. This is pure biochemical violence. The cascade continues until you activate C5b. C5b then recruits C6, C7, C8, and a whole ring of C9 molecules.
And they assemble into something.
They literally self-assemble into a physical tube or a pore. They punch a hole right through the bacterial cell wall and membrane.
It drills a hole in the hull of the submarine.
An unstoppable one. And water rushes in, osmotic pressure takes over, and the bacteria swells and pops. It's called lysis. It's a direct kill.
It's just incredible to think this is happening in liquid form. No cells required, just proteins floating around, assembling, drilling, and killing.
It perfectly illustrates the point of this whole deep dive, doesn't it? Blood is not passive. It's not just red water carrying cells. It is a highly regulated, chemically active, living organ in liquid form.
Let's try and wrap this all up. We started with albumin sucking water back into our veins like a sponge. We went through the armored trucks of transferrin, the recycling centers of the macrophage, the smoking gun of lung destruction, and finally this heavy weaponry of antibodies and complement.
The takeaway for me is the theme of balance. Everything is about exquisite regulation. Albumin balances water. Hepcidin balances iron. Antiproteinases balance destructive enzymes. And when that balance tips, either due to genetics like in hemochromatosis or an environmental insult like smoking disease happens.
And that leads us right to our provocative thought for the day. You mentioned it earlier, the anemia of inflammation.
Yes. Think about this next time you look at a lab result or you hear about someone being treated for a disease. We often see a number that's out of the normal range, and our first instinct is to fix it. If a patient with a chronic infection has low iron, our instinct is give them iron pills.
But the body is lowering the iron levels on purpose.
Exactly. The body is actively and intelligently shouting, hide the food from the enemy, to starve the invading bacteria. If we come in and blindly supplement with iron, are we actually helping the patient, or are we feeding the infection? It raises the question, how many symptoms that we treat are actually sophisticated defense strategies that we might be clumsily interfering with?
That is definitely a thought to chew on. The body, most of the time, really does know what it's doing.
Usually. Until it starts misfolding proteins or deciding that cat dander is a mortal enemy. But yes, for the most part, it's a brilliant machine.
Well, I will never look at a simple paper cut the same way again. There is an entire universe of complex machinery leaking out of there.
A universe of infrastructure and defense, all in one drop.
Thanks for diving in with us. Check your iron, appreciate your liver, and we'll see you on the next deep dive.